Tonight it's my pleasure to introduce to you Vatsan Raman. He's in the biochemistry department. He grew up in India, then he came here to go to what used to be the Missouri School of Mines in Rolla, but as he pointed out, it's gone through 12 or 13 different name changes. All right, just 2, but hyperbole. And now it is called the
Missouri Science and Technology University.
Very good.
Let's see if that works for them.
Then where'd you go for your PhD?
Seattle, University of Washington, the other UW.
There are 2 UWs?
That's right. Wow. We're not the center of the
world. I just knew about the one in Wyoming. 1 Mississippi, 2 Mississippi. God. Okay. And Vatsan's been here for the last 2.5 years. He's in the biochemistry department. He's going to talk with us about synthetic biology for biomanufacturing, the environment, and human health. This is a little out there for me. I'm looking forward to hearing some of the stuff that's different than what I'm used to. I know a little bit about chemistry. I'm wondering what synthetic biology is. Would you please join me in welcoming Vatsan Raman to Wednesday Night at the Lab.
Can folks hear me okay? Is it good? Okay. Well, thanks, Tom. Thanks for the introduction. It's a pleasure to be here. So I'll maybe, so again, the topic of my presentation is synthetic biology, but we'll cover a few different applications and in biomanufacturing, environmental sustainability, and human health. And so what I thought I'll do is sort of repeat what Tom said and maybe give you a sense of my background and sort of what led me to where I am here. So I got my degree in chemical engineering from India, and I was an intern in a petroleum refinery, and then I was a salesperson selling these large pumps and control valves in this company, Flowserve. Then I sort of really missed all the technical stuff, and then I went to, again, the Missouri University of Science and Technology, that's the current name, and I got my master's in chemical engineering. And I got my PhD in computational biology at the other UW, University of Washington in Seattle. And I was a postdoctoral fellow at Harvard for about 4 or 5 years. Okay. So just sort of quick show of hands, how many people have heard of the term synthetic biology before? All right, that's great. So what we'll do is we'll start with the Wikipedia definition of what is synthetic biology. It is designing and constructing biological modules, biological systems, biological machines, or the redesign of existing biological system for useful purposes, right? Now I have way more slides than a 1-hour presentation. What we'll do is we'll just make a hard stop at an hour, but really feel free to stop me at any point, and it'll be sort of fun if it's a back and forth discussion, right? Okay, so this is the textbook definition of synthetic biology. And what this means practically is the idea of how can we use sort of synthetic tools in biology to really sort of fulfill human needs. That's sort of one way of thinking about it, right? And you can ask the question, can we use these design and engineering principles in biology with biology at multiple sort of scales, right? So one application could be at the scale of, say, tissues and organs, right? Say, for instance, for regenerative medicine, right? So the idea would be to synthesize organs like almost in a petri dish, and that's already happening now. The other scale would be at the scale of, say, a community of microbes, right? And so the application here would be to sort of understand how antibiotic resistance has emerged, or maybe even understand how metabolism works, because our gut is full of microbes, right? You can think of synthetic biology at the scale of operons or genomes of organisms, and that's the place where you would tinker to get these organisms to make, say, chemicals and fuels. And you can think about it at a molecular scale where we want to modify biomolecules like proteins and RNA to make therapeutics and biomaterials, right? So that's sort of many different scales. And I was sort of really drawn to this idea of how we can use microbes as chemical factories, right? And sort of the big picture here is how can we really use biological-based production to completely supplant or replace, our sort of current fossil fuel paradigm, right? And so there are a few different sort of motivations for why we might want to have microbes as chemical factories. One would be, again, like I said, sustainable production, right? And so the idea here is how can we replace petroleum or petrochemical-based products using biological manufacturing, right? The second motivation is this idea of precise stereochemistry, right? So drug molecules or drug-like molecules have sort of really an exquisite arrangement of atoms in what's called a sort of a stereochemical property, right? And enzymes are really good at making these stereochemical molecules, but if you did chemical synthesis in a traditional sense, that's really hard to do, right? And so biology is really good at making precise molecules very efficiently in a way that sort of traditional chemistry couldn't, right? And the third, and perhaps personally the biggest motivation, is this question of what is the unexplored chemical diversity, right? So, all the molecules that we use, from our toothpaste to detergents to, really everything, is really a very small fraction of the chemicals and metabolites in the biosphere, right? So we've sort of barely scratched the surface. And so, for instance, if you can ask the question, well, do we know all the metabolites in a marine bacteria or a soil bacteria? And these could be the next new set of therapeutics or smart materials, right? And we really have no clue, right? So what we use now is really sort of scratching the surface. And so there's this whole unexplored diversity of enzymes and molecules that we could discover, which could be sort of really impactful. So these, to me, sort of big motivations for why we want to do this. Now, so here is a sort of a snapshot of what is the size of the bioeconomy in the United States, right? So this is about $350 billion or $400 billion. It is sort of split into GM crops, so this is corn and all that stuff. And then you have biologics, and so these are proteins that are used as therapeutics. So antibodies, for instance, that are widely used against cancer would fall in this group. And then there is the industrial biotech group, right? And so this is the focus of my presentation, and you could sort of further split this into food and ag, and then biopharma. So this is sort of your traditional small molecule drugs, right? Fuels, so biofuels or biological replacements of petroleum fuels, and fuel-based or biochemicals, right? So this is sort of the split. And so the size of this market is about $130 billion, right? And so to put this in perspective, right, the size of the petrochemicals industry in the United States is about $800 billion, right? And so if we really want to have a vision of replacing petroleum-based products with biology-based products, I mean, there's a lot of ground to cover, right? And so one can then ask the question, how is biological engineering different from traditional engineering? So in traditional engineering, right, so think about this. If you want to make some kind of an integrated chip, right, so in electronics, so what you would do is you would maybe come up with a handful of design based on some rational principles, and then you would maybe make 5 or 10, test them out, see what works best, and then that would be your sort of design component, right? But that's not the way biology picks the best solution, right? What biology does is it evolves. It uses evolution to really pick the winner. And so that in perhaps is the most significant difference between traditional engineering and biological engineering, right? And in that we can harness the force of evolution to pick out the best, say, design or the best product. And so let's sort of dig a little deeper here, right? And ask the question, if you wanted to engineer, say, a microbe to produce some useful biofuel, right? Then there are really sort of 3 steps to this. And this is true of any engineering, but we'll look at this in the biological context. There is the design step, there is the build step, and there is the test step, right? Now, we're at a point where we have really amazing software tools and other methods to come up with many prototypes, right? So we can come up with many different versions of a pathway, many different versions of the genetic arrangement inside a cell, right? So we can do this, so there are computer-based systems where we can design or come up with many design prototypes. And just to put a number, this is, 10 to the 8th or 10 to the 9th different designs can be done fairly easily on a computer, right? Now, we've also had sort of really amazing advances in the ability to synthesize DNA. And this is important because sort of the most fundamental part in biology is the DNA, right? So if you come up with designs, you ought to have DNA that reflect those designs or are those designs. Now, this was a huge bottleneck even until, say, 5 to 10 years back. And now we've had some amazing advances in the last, I would say, half a decade or a little more that allow us to build these designs in exceedingly large scales, right? And so now you have the design step, which is fast. You have the build step, which is, again, very large. Now the question is, well, you come up with all these designs of microbes and you build them, but we don't know which one is working, right? And so that is the huge bottleneck. So just sort of imagine going from 10 to the 9 potential entities that you've built and only being able to test 10 to the 3 because we have analytical methods which are really low throughput, right? So that's a huge bottleneck if you think about how sort of biological engineering is working, right? Now, just sort of another way of sort of framing this sort of argument is what are the big technologies in the last, say, maybe 10, 15 years that have really changed how we can engineer microbes? One is this idea of next generation sequencing, right? So this means that we can sequence large volumes of DNA of sort of bacterial genomes. And what this has helped us do is sort of catalog all the genes in many, many kinds of bacteria, right? And so now we know what their genetic makeup is. The second enabling technology is this idea of metabolic models. And I sort of alluded to this in the previous slide, where you have computers that can generate. So again, if you just imagine a cell, it's a really sort of complex bag of biochemical reactions happening, right? And you ask the question, well, I want to make a specific product. How do I rewire the metabolism of the cell to get it to make what I want, right? And there are many, many ways of doing it. And there are computer programs that can sort of move things around and come up with potential solutions, right? And again, the third advance, like I said, was sort of the DNA synthesis and the idea that we can use new methods to modify DNA. So one example is the CRISPR-Cas9 system. Have folks heard of CRISPR-Cas9? Right. So that's sort of one major advance, right? And again, now the final question is, well, who is the winner, right? How do we evaluate how they're performing, right? So that sort of is the rate limiting step. So I'll just sort of quickly run through the slide. It's sort of the same argument. Now the question then is, well, why do we need to evaluate 10 to the 6 or 10 to the 7 different cells? Why do we even need to do that? In traditional engineering, we just do 5 or 6 or 10, right? And so I'll give you sort of one sort of cartoon example, and you can sort of imagine the scale of the problem, right? So now imagine you have a certain input molecule, right? And you want to convert it to some final product that you care about, right? And let's say there are 4 or 5 genes involved in a pathway. So these are all enzymes that convert this to some product and then some other product and some other product, and then finally, boom, you have the end product, right? Now, the objective is to maximize what is called as the flux. And that is basically nothing but stuff through the pathway, right? You want to maximize the carbon that goes through the pathway so that all the input reactants get converted to a product, right? Now, every enzyme performs differently. Every enzyme is made at different amounts inside a cell, right? And so now if you imagine each of these arrows, the width of the arrow being some measure of how much stuff it can sort of carry forward, right? You can look at this figure and you'd say, well, this is the bottleneck step, right? So these guys are going fast or can handle a lot of carbon flux. But this guy is the wimpy enzyme here, right? And then what you might want to do is do some sort of genetic jiggery-pokery and then see if you can increase the flux through this step. Right? So that'll be sort of one way of, sort of one engineering step you can imagine. Now, conversely, you can again imagine the same pathway, but then you might have something that gets siphoned away, right? So you might have a side reaction that says that takes this intermediate and puts it in a different pathway, right, which is undesirable, right? And now what you might want to do is really sort of throttle this step, right? so that you, again, maximize the flux through this pathway to make high amounts of the product. So it's, a simple way of imagining this is just like a complicated piping system, right? And so you have your feedstock that goes inside a cell, and then you have end product, and there are many, many connections of pipes, and there are many, many valves that are turned to different levels. And what you're trying to do is push as much through this pathway to get as much out in as short a time. Right? So that is sort of the objective. Well, I won't get into the math here, but I'll just make the point that if you take a simple pathway of 10 genes, which is sort of a normal-sized pathway in any organism, the number of combinations you have to test, right, where you say, like I said, optimize the flux through the pathway, really runs into the millions, right? And so this number is, the combinatorial space is very, very large. Okay, so this is sort of the problem statement. That's why we cannot pick 4 or 5 and test them. We really have to test a large number to see what works. And in some ways, this also reflects our sort of lack of knowledge, right? So if we knew exactly what to do to get them to make your biofuel, we would exactly make that design, right? It's because we don't know enough, we have to try many, many different combinations and see what works. So now the question is, well, if you have 1 million candidates to test, it's just not feasible, right? You cannot pick 1 million different bacteria and test them in a lab. It just doesn't scale that way, right? And so now we come back to the question of, how can we use evolution? Is evolution our friend? And can we harness evolution as a force to help us pick the winner? Because that's what really sort of natural evolution does day in and day out, right? Now, the problem with this approach is that most of the molecules that you might want to make are not essential for the bug, right? The bug has really no interest in making biofuel for you. It just wants to multiply and be happy, right? And so you're sort of forcing it or coercing it to make this, and really it's not essential for its survival, right? And so evolution only works if you can somehow tie it to survival of that species, right? Or fitness of that species. Right? And these molecules, really any molecule that you want to make is not essential for any bug. And so now we have a problem, right? So how do we turn this into, if you want to use evolution, you have to turn this into a fitness for the bacteria. Does this make sense to people? Yeah, okay. All right, so what we work in my lab is to come up with these ideas where we can have this artificial evolutionary coupling. And the way it works is as follows. There are these proteins which are called allosteric transcription factors. It doesn't matter what the name is, but the idea is to use them as a biosensor, right? And so the way this works is as follows. So now imagine a protein, right? And so this protein sits on DNA, right? It has 2 domains to it, or 2 regions to it. One region that binds to DNA, and the other region that can bind to a small molecule, right? And now let's say that small molecule is the molecule that you want to make, say like a biofuel molecule, right? Now the way the protein behaves is the moment it binds to that small molecule, right, say a biofuel, it just comes off of DNA, right? Now this is a super valuable property because now what we can do is we can put a gene which is an antibiotic resistance gene, right? So this is essentially an antidote protein which allows cells to survive antibiotics, right? And so the mechanism is follows, right? When the protein sees the biofuel molecule, it comes off of DNA and then it makes this antidote protein, right? So now what we've done is we've created this artificial system where the production of this biofuel-like molecule is required for the production of the antidote, right? So we've created this artificial coupling. Does this make sense? Yeah? Okay, so this is sort of the most complicated step. Everything else is easy after this. And now once you establish this, right, you now have chemical concentration is now coupled to cell growth, right? And so the way your lab evolution experiment is going to work is as follows, right? So imagine some starting bacteria that you want to use to produce some useful molecule. you can use CRISPR-Cas9 or any number of methods to genetically diversify the population, right? So now you have a large population of cells, each carrying some mutant of a pathway that you care about, right? Now the question is, who is the winner here, right? And so let's say one of these cells is the jackpot cell, right? And it's making tiny amounts of the molecule of interest. Now what you can do is you can now throw antibiotic at the population of bacteria, right? And because this guy will trigger the production of the antidote, this guy will survive, right? And everybody else will perish, right? So this is selection for you, right? And you can go back and iterate this process many, many times, really, sort of genetically diversify, select, genetically diversify, select, and ultimately you can pull out a winner, right? And so this turns out to be a sort of a generalizable strategy that one could employ for the production of all kinds of molecules. And the reason for that is these proteins are diverse, they're ubiquitous, that you can find them in all kinds of bacteria, and they bind to many, many types of molecules. And so the strategy is generalizable, which is important, right? It cannot be a one-trick pony. You have to be able to apply the same strategy to produce many types of chemicals, right? So I'll give you sort of one example here that we demonstrated a couple of years back. Now it is to produce this molecule called naringenin. Now naringenin happens to be, it's called a flavonoid molecule, but essentially it's found in grapefruit and oranges and tomatoes, sort of like a pigment-like molecule, right? And it's known to have all kinds of sort of medically useful properties. It's an antioxidant, it has anti-inflammatory properties, etc. And so what we said was naringenin is not made in E. coli, so let's get E. coli to make naringenin for us, right? And so without getting into too many details, there are two precursors for this. One is malonyl-CoA and the other is tyrosine. And you put them together. Again, there's a bunch of E. coli genes involved in the pathway. And there are a couple of yeast enzymes and a couple of plant enzymes. You throw everything into E. coli. In principle, it should make naringenin, right? Now, but of course, in practice, it does not because the system is not optimized for production. And so what we did was we looked at the metabolic chart of E. coli, and this is a really, really simplified version of that, and then we ended up picking like 17 or 18 different genetic targets, and we said, all right, these might be the most important ones to produce naringenin, and then we mutagenized them, and then we had a large population of 1 billion different cells, each having some combination of mutations. And then what we showed was over 4 rounds of evolution, we could really bump up the production 36-fold. Right, and I think to me this was sort of insightful because this is really sort of metabolic engineering for dummies, right? And the reason I say that is, I have some vague idea of what's important, but I don't know the details, right? And I'm really just throwing the kitchen sink at the problem and letting evolution tell me who the winner is, right? And so you have an excuse for not knowing enough because it still works, right? And then what we did was we put it into a bioreactor. So this is done in sort of lab smallish scale, and then you can scale up the production. And this was the highest naringenin produced directly from glucose, right? And so the idea is that this strategy actually works. And so one vision for how one might do biomanufacturing is to have a workflow like this, where you use these genetic diversification methods, create a really large number of prototypes, if you will, use the biosensor to really cut down this population of 1 billion different solutions to maybe a few hundred solutions, right? And then you can use more precise methods like mass spectrometry to then pick maybe 100 or so potential hits, and then you scale them up into a bioreactor, and one of them, or maybe many of them, would be winners, right, that one could put in a bioreactor. So I'm going to switch to another topic after this, but do folks have any questions about this so far? Yeah.
So basically, for every molecule you have to make, you have to know enough about the pathway to do the mutagenesis specifically of those genes with CRISPR or whatever.
That's correct. Some a priori knowledge will be really helpful because then your search is a little more focused,
Yeah.
right? But you don't need to know the exact solution. All right, so I'll then go to the next topic. And again, so we can use these biosensors in many different contexts. I've mentioned one in the pathway engineering. We'll discuss this topic next, which is this idea of functional metagenomics and how we can use this for bioremediation. But something that we're really interested in is to sort of turn these protein sensors into paper-based sensors, like pH-style sensors. And the idea is that then you could deploy this, say, in some remote location for like measuring a bunch of metabolites that are, say, polluting a water body, for instance, right? And so it would be, so the way it's done now is then you have to take samples, you have to ship it to some lab, and then they take a couple of weeks, and then you get the results back. But the idea is that you can then use these protein-based sensors, you put them on a piece of paper, and there's some more tricks that you have to do to it. But the idea is that then you can take this piece of paper to some remote location, drop a sample, and see if it changes color. And you can have a panel of these sensors that will allow you to measure a bunch of different, say, conditions, right? And again, there are other applications to this. Okay. So have folks heard about the term microbiome or microbiota? Mm-hmm. Yeah. So it's really cool because we now know that we're not, we're a collection of human cells and microbial cells, and these microbial cells live in communities inside us, and it's not just us, they live in communities really everywhere. And it turns out, there are really sort of many number of fascinating properties of these microbiomes. But one that is specifically of interest to me, and I mentioned this early on, is this idea that these microbial communities are really a treasure trove of enzymes, right? You have these large collections of microbes with really sort of amazing metabolic capacities, and they get that because they have enzymes that can do really cool chemistries, right? And so what if we can lay our hands on those enzymes, and how can we use them, right? Just to give you an example, again, if you have a collection of microbes, say, that you obtain from soil, these are microbes that are exposed to pesticides, and so they have enzymes that can chew up pesticides, right? You can get microbes from the human gut, and you can investigate proteins in them that are involved in drug metabolism and how fast drugs are consumed or metabolized in our bodies. So the cow rumen, it turns out to be a really amazing resource for enzymes that can chew up cellulose, right? Because cows eat that stuff. And so they have microbes that have these enzymes. And the reason that's important is because the sugars in cellulose are really trapped in a state that's not available to us, right? And so if you want to make biofuels, for instance, you really have to free up those sugars, and the enzymes in cow rumen do that, right? And so again, just sort of emphasize the point that it's a rich treasure trove of enzymes. Okay, so just to sort of again give you some numbers here, right? To illustrate the point that the diversity that we have out there versus what we know now, right? So 1 gram of soil, right, is estimated to contain 10 million to a billion prokaryotic cells, right? So just 1 gram of soil. Now, which effectively translates into 1,000 to 10,000 unique genomes in 1 gram of soil, right? So we have one human genome, and so we 1,000 to 10,000 different types of genomes in 1 gram of soil. Now, the total number of genomes that we have in the database, right, and sort of is close to 100,000, right? So what we have is essentially like 10 to 20 grams worth of soil DNA in our databases, right? And so just think about that number for a second, and how much diversity is out there and how little we know, right? And so the number of unknown enzymes is just staggeringly large, right? Now, there's obviously no straightforward way of figuring out what these, enzymes do, but you have to start somewhere, right? And so I'll give you again a couple of instances of how. So sort of and this idea is called sort of metagenomic mining, which is how do you mine these genomes to again, harvest new enzymes. And so industry uses this all the time, and they've had a number of examples where they've been successful. And so this is just a chart that shows you all the kinds of enzymes that, that industry has identified. So these are lipases, amylases, proteases. So for instance, these are used for production of polymers and detergents, and they're used in the food industry. And so again, there's high value in obtaining these enzymes. Now, there are two ways people go about identifying these enzymes. Right? One approach is called the sequence-based approach. It's not sequence-based, it's sequence-based. And the other is function-based. Right? So what does this mean? So the idea with the sequence-based approach is, again, you go collect soil from some location. You can harvest the DNA, just sort of extract the DNA by simple experimental techniques, and then you can send it out for DNA sequencing. So have people heard of like Illumina sequencing and all that stuff? Like, right? So there's just large volume sequencing. And what this does is you just sequence the DNA that's present in the soil sample. Right? Now what you then do is once you get the sequences back, then you can ask the question, do these sequences resemble enzymes that we already know in our other databases? Right? So thinking is that if the sequences resembles, then it's likely to have the same or similar function. Right? Now, of course, that's a bit of a chicken and egg problem here, right? Because you're going out to look for new enzymes, but then you're trying to compare them with existing enzymes, right? And so if you have an enzyme that doesn't match any known sequence in our databases, then that's just, you have no clue what that enzyme does, right? And so the way I think about this is that that's a huge limitation of this approach in that you keep looking for stuff that you already, it's the unknown unknowns that's important, right? Or the known unknowns in Rumsfeld speak, yeah. And so the question is, so this approach has its limitation. Now the other approach is this function-based screening, and the way this works is you can. So let's say you take an example of some pesticide that you've applied somewhere, right? And you ask the question, give me the enzymes that can degrade this pesticide, right? And so what folks would do is you can take that pesticide, put it in some solid plate, like a regular sort of bacterial plate, grab bacteria from soil, sort of slap it on the plate and see what grows, right? And if it grows, it means that it has the full metabolic capacity to take that pesticide and essentially consume it and use it as a carbon source, as a nitrogen source for growth, right? Now, to me, the problem with this approach is that you're screening for the, there are two sort of major issues with this approach. One is that it requires the full metabolic capability of the organism to consume that molecule, right? So you're looking for a specific chemical transformation, but what you need is that enzyme to be able to convert it into a product, and then to another product, and then to another product, and finally to something that the bug can use as a carbon source or nitrogen source. So you're really asking too much, and hence your solution, the number of bugs that you might find will be smaller, right? The second problem, in some ways is the bigger problem, is that the vast majority of bugs that you get from the environment will not grow under lab conditions, right? And so that number is like 5% or 10%. So the remaining 90% won't even grow in the lab conditions. So again, you're losing a lot of potential solutions by using a culture-based approach. And so I'll maybe mention one more limitation. So in some cases, what you can do is you can do these screens where you look for some color change of some enzymatic activity. And you can say, well, if this changes from nothing to red color, maybe there is an active enzyme that does something, right? Now here is the number. And so the way this experiment would work is you would pick bacterial cells into a well, right? And so each plate has like 96 wells, and you have to screen hundreds of thousands of wells to find something that changes color, right? And again, just to illustrate that point, so again, this is company data. They had to screen 900,000 different cells one at a time, right? Using robotics, but it's still one at a time to find 15 hits, right? So just imagine a poor graduate student picking, bacterial cells one after the other, and it's just that the success rate is just really bad. Now the companies can do it because if they find a winner, they can, there's a lot of money to be made, right? But it's just not it's not elegant, B, it's not scalable, right? And so what we're trying to do is this idea of, again, we've shown that we can use these biosensors to screen like 1 billion different cells, and we're not picking one at a time. They're all in the same pot, right? It's just we let evolution pick out the winner. And can we use the same strategy to identify new enzymes? And the way we do it is, again, we get the genomic DNA from, say, soil sample, and then we essentially put those into cells, but we don't separate the cells. We just let evolution tell us which cell is the winner, and then we can pull that out. And so I'll give you sort of one example. There are two examples, but we're specifically working on one. So we're working on identifying enzymes that can degrade what are called as anthropogenic chemicals. And so these are chemicals that were introduced in the environment in the last 50 to 60 years, right? And so they're totally synthetic chemicals. And what's happened is bacteria have adapted machinery to be able to use or chew up these molecules. So one example is this molecule called Pentachlorophenol. It's a really bad molecule. It's used as herbicide, pesticide, and all that. It's high toxicity. Its bioremediation rate is fairly slow, and it's actually banned in some countries. The other molecule that we're working on is this molecule called atrazine. It was, up until some time, the second most widely used pesticide in the United States. It's been detected in drinking water. It's called an endocrine disruptor because it mimics some of the molecules of the endocrine system, and so it sort of messes up our endocrine system. So what we decided to do was we said, all right, atrazine. There is one bug. It's called a Pseudomonas. It's the name of the bacteria that can chew up atrazine and make this intermediate molecule called cyanuric acid. Right? Now, cyanuric acid by itself is benign, right? It's not toxic. It's in fact one of the chemicals that is used in swimming pools, right? So it's totally okay. But this molecule is not a carbon or nitrogen source because most bacteria don't know how to handle this, right? Then they need additional enzymes that would ultimately convert it into ammonia, and now they're good to go, right? And so we said, well, if we can take atrazine to cyanuric acid, and that's a big part of the problem solved, right? Because we've turned a potentially toxic molecule into a benign molecule. And so now what we're working on is designing a biosensor that can specifically sense this so that we can then do our large-scale environmental screening to identify new enzymes. And what we now actually have is a biosensor that works on cyanuric acid. So here's just data that shows that it's responsive to cyanuric acid but does not respond to atrazine, so which means it is specific to that molecule. And what we've then also showed is that if you drop atrazine into the bacterial culture, but then give the bacteria these 3 enzymes, it'll convert it into cyanuric acid, then it'll trigger the sensor, right? So at this point, we're primed to get in environmental samples. So this is the control system, which should work, and it does work. And so we're now at this point primed to actually get environmental samples and do the screening. So we haven't done that yet, but that's our next step. How much time do we have? Okay. I'm going to make yet another pivot. Are there any questions? Yeah.
When you were talking about the two ways of doing things, one of them was you said you could only annotate the same
Known stuff, yeah.
Why?
Well, because you...
Can you repeat the question, please?
Yeah, so the question is, why does the sequence-based approach result in annotation of or cataloging similar sequences, right? Am I paraphrasing your question okay?
Well, why can't you do the unsimilar ones?
Well, so when you get the DNA from a soil sample, right, you sequence it, you have to, it's just a bunch of letters, right? You have to say, what does it match known stuff? What usually happens is about 15% or 20%, no more than that, have any remote similarity to known sequences. If it's an unknown, if it doesn't resemble anything that's known, you have no clue what it does, right? So you just have to discard it completely because you cannot even infer what the enzyme does. But if those sequences resemble, and so the idea is that then these sequences are evolutionarily related, if they have some match, and if they're evolutionarily related, they're likely to have similar function. That's the argument. Now, it holds to a certain extent, depending on how close the match is, but sometimes similar looking enzymes do completely dissimilar reactions. Any more questions? Okay, so we have about, I guess, 10 more minutes, and I have like 60 slides, so I won't do 60 slides. So what I'll do is I'll maybe do a hard stop in like maybe another 7 minutes or so, and we'll just leave the floor, have the floor open for questions. Okay, so this is a problem that we've started working on for the last couple of years now. It's this idea of how do we deal with resistance to antibiotics, and I sort of mentioned this early on, right? Now here is a study. Sorry, I don't have the citation for this, but the idea is that this is the projected number of deaths from antibiotic resistance, right? And so again, what that means is bugs are becoming increasingly resistant to the antibiotics that we already have, right? And it's projected by 2050, this will be a killer that's bigger than cancer, right? And so it's a big problem that's brewing, and where we have to find a solution to this, right? Now, this chart shows you when an antibiotic was introduced, right? And when the first resistant strain was found, right? And so if you look at penicillin, it was introduced in 1943 or whenever, 44. And then you had the first resistant strain in about 20 years, right? And this is tetracycline, erythromycin, gentamicin, vancomycin. It's about, so 10 to 15 years is how long it normally takes for some strain to become resistant to that drug. Now, but you see these molecules, right? The moment you introduce the drug, you have a resistant bacteria that's waiting for it, right? And the reason for that is the bacteria have picked up sort of genes that allow them to not become susceptible to these molecules, right? From other molecules in the past. Right, so they're all primed and ready to go, right? And all that they have to do is just have a few mutations here and there, and then, lo and behold, they're resistant to the next antibiotic, right? So this is a serious problem. Now, sorry, you can't see the numbers here, but what this chart shows you is the number of drugs that are coming that came through the pipeline at different points of time, like this. So this is 1980 to 84, 85 to 89, all the way to 2010 to 2012, right? And what this tells you is our pipeline is running dry, right? So we don't really have too many. So we have two problems. One problem is that if you have the molecule, the resistant bacterium emerges pretty fast. Well, and the second problem is, well, we don't have molecules, right? And so and so I think as you sort of look at this, I mean, of course, it's not all dark and gloom. I mean, there are last resort antibiotics that are being used, but we have to find a sustainable solution to this. And so again, this, I said, well, we have to sort of think of fundamentally new way of solving this problem, and really sort of old is the new, right? And so we can ask the question, well, how have people sort of thought about this problem 50, 70, or 100 years back, right? Or revisit old solution, which are bacteriophages. Have people heard of this term called bacteriophages before? Okay. So bacteriophages are viruses, but so the viruses that we commonly refer to as viruses are viruses of higher organisms. But bacteria also have viruses, and they are called bacteriophages. And what it means is it's called it translates into a bacteria eater. And it is the most abundant life form on Earth, bacteriophages are. It's estimated that there are 10 to the 31 bacteriophages in Earth. It's just an unbelievable number, right? And it's 3.8 billion years of evolution, and in some ways one could think of this as nature's most successful evolutionary experiment, right? Because they've been around for forever. Now, bacteriophages, or also called phages, have an illustrious history. They were discovered in 1915 by Frederick Twort, and he had these glassy, transparent spots on a plate, and what we now call as plaques. Felix Darel and Eliava and others realized the therapeutic potential of phages. And so phage products were sold in the 1930s. Delbruck, Luria, and Hershey and others studied these T family phages. They ended up getting a Nobel for that. And Jacob and Lederberg and Esther Lederberg studied these lysogenic lambda phages. And so much of the genetic regulation that we know is from phages. Phages have been part of multiple sort of landmark, scientific landmarks, if you will. The first restriction enzyme was isolated from phages. Right? So if you've done molecular biology, restriction enzymes are really very important in molecular biology to cut and paste DNA. The first RNA sequencing was of phage MS2. Right? The first DNA genome sequencing was the phage 5X174. Phage display is a method that's again widely used to display proteins for all kinds of applications. The first synthetic genome, which is sort of a genome built by chemical DNA, not biological replicating DNA, is a phage genome, and it works. So you put this together and it actually works as a phage. And CRISPR-Cas9 again is a mechanism that bacteria have evolved to fight back phages. So that's how we know CRISPR-Cas9. Now let me just quickly check time. All right, I'll maybe do 5 more minutes and I'll stop after that. So what's happened is, the phages were sort of really a big deal in the 70s and 80s, and over time they sort of fell out of favor, and there's not enough funding in phages. And that field sort of, I wouldn't say it died down, but there hasn't been much activity for almost 20 years. And now we can then ask the question, well, why bacteriophages and why now? And the reason is there's sort of a renaissance that's happening in bacteriophages. And the way I think about this is really the confluence of what bacteriophages can do and the synthetic biology tools that we have at our disposal now really opens up sort of many exciting research avenues. Right? And I think it's sort of that perfect storm of what phages have the ability to sort of kill bacterial cells, and all the tools that we now have at our disposal, which we did not have 20 years back, to modify and manipulate these phages. And so in terms of application, again, genome engineering and evolution, we can sort of think of sort of getting genetic parts that one can use, phage-based therapeutics. You can use phages to deliver a genetic payload. So for instance, if you wanted to modify your microbiome, and so just one example would be, let's say you're traveling to some place where you may be susceptible to certain kinds of ailments, then you can sort of essentially modify your microbiome by delivering genes to your microbiota so that they can sustain whatever onslaught that they'll have in a new environment. Right? So genome mining for new therapeutics, and then you can use diagnostic tools. So phages are being used in food safety. So because phages can specifically bind and detect bacteria, you can use phages to, it's used in the food industry to check what is the contamination level. Okay, so I'll skip this. We've known about phages for over 100 years, and these are publications that came out, well over 100 years. So I'll just make this one point. There were sort of the phages were initially commercialized in the 1920s and 30s. And so Felix Zarel had, got £1 million to sell these bacteriophage medications to treat different kinds of disorders in this laboratoire bacteriophage. And these were essentially, sort of phage capsules that were, sold as antibacterial treatment. There were really no quality controls or standards, and some of them worked and some of them did not work, and so it eventually did not, was not a huge commercial success. And again, this is a picture from 1936, so these are the different phage-based products that they sold for all kinds of ailments back then, right? And it turns out that this laboratoire bacteriophage is now L'Oreal. So if you're using the cosmetics from this company, that's the illustrious history that they have. Okay, and so then we found penicillin, and then it was a miracle drug, and it worked amazingly in the war, and then we never looked back, and we never had to use bacteriophages, right? But of course, that's now changed, right? So I'll maybe end with this slide. Well, maybe one more slide just to highlight the challenges. Now, phages are sort of in the way I think of phages as the ultimate smart drug, because they're highly precise. So if you consume an antibiotic, it will really kill a lot of different things in your gut. But phages are very precise and will kill specific targets, and so will sort of keep the rest of your beneficial community intact. Now, phages are evolvable, and I think this is an important trait, because once an antibiotic becomes useless, there's not much you can do with that, right? But phages are biological entities, right? So if the bacteria will become resistant to a phage as it becomes resistant to a small molecule, but what you can do with the phage is go back and do some genetic tinkering and keep it a couple of steps ahead of the bacteria, right? And that's what we do with the flu vaccine anyway, right? And so we stay a couple of steps ahead of the flu vaccine just by sort of modifying a few different components, right? So because we have the predictive power to know what the next strain might be. But you can't do that with small molecules because it's a dead end, right? And phages are auto-dosing. And this is sort of an interesting property, because the way the phages work is they get into the bacteria, and once they get in, they make many, many copies of themselves, right? And then they sort of crack the cell open, and then they go infect the next bacteria, right? So all that you then need is a smallish dose, because it's auto-dosing, because there's this feed-forward loop that every time it infects, it sort of multiplies. So there was this one case which really sort of caught a lot of attention last year. It was this professor from UCSD, UC San Diego, and he was traveling with his wife in Egypt and was infected with Acinetobacter baumannii. They did not know what it was, and he was just given regular antibiotics, and none of it worked. And then he had to be airlifted to Germany because his health was sort of deteriorating very quickly. And then he was airlifted to San Diego. Now, it turns out his wife was also a professor. And so they gave the regular medication course, and it was not working. And then she started looking for sort of alternative approaches and found that there was a group in, I want to say Belgium, who had phages against Acinetobacter. And there were a couple of groups in the United States. So the Center for Phage Technology in Texas A&M, College Station, and the US Navy research labs, they also had a collection of phages. And so now, typically you could not do this without FDA permission, because the drug has to be approved. But the FDA has a mechanism which is called as compassionate use. So you can make a one-time exception if all the other treatments are failing, and so they let you use a trial medication. And so they got compassionate use approval, and then they applied the phage, and he started recovering after the phages were delivered. He was okay for a week or two, and again, then the bacteria became resistant to the phage, and his health condition went south again. And then they took the resistant bacteria, went back to the lab, re-evolved the phage to go back and kill that resistant bacteria, and now he's chill now. He's okay, right? And now, so this is, no, the problem, of course, is this is not a scalable model because you cannot do this for every patient who has a certain slightly different type of sort of pathogenic bacteria. But the idea is that can we turn phages into a more of a general product that people can consume against antimicrobial resistance. I think it's almost 8 now. I'll stop, but I'm happy to answer more questions.
So in the case of this phage therapy, is there issues about distribution, body distribution? Does it get everywhere?
Yeah, no, so there's a lot of, well, okay, well, thank you, because I can go 2 slides more. Okay, so there are a few challenges. One is that, so these are topically applied phages. So we still haven't done the intravenous phages yet. Well, I mean, people have done studies. What we don't know is what the immune response for intravenously injected phages would be. So if you had a topical infection, you can throw a ton of phages at them and nothing's going to happen. Now, we already have like 1 trillion phages in us, so it's not like this is foreign to us. But what is different is, there is the chance of immune response if it is delivered into the bloodstream, right? And so people are figuring out how that would work. But again, in his case, I think it was just topically applied, where you just throw the, bottle of phage, perhaps more sophisticated, but yeah, you apply it to the wound or wherever the infection was. Yeah.
So, just so I understand, a phage is a naturally occurring bacteria eater. Is that what you're saying?
Yeah.
And there's some extremely large number of them.
Mm-hmm.
And is it trial and error that you find phages and you say, oh, this is a bacteria, let's try them on this, and they either work or they don't? I mean, they're all different, they're not just a lot of phages, they're all different kinds of phages.
Yeah,
right?
yeah.
And so only some phages work on some bacteria, and maybe they've already evolved to work on the next level of the bacteria if you ever get those two together. Is that what's going on there?
Well, so it's really complicated because, it's, I mentioned the specificity of phages as an advantage, but you can also think of it as a disadvantage, right? Because, so imagine, again, 2 people have slightly different strains of a bacterium, right? A phage that might work against one strain may not work against another strain. So that's a problem because how do you do drug development then, right? And so what's happened, so the issue is that they are specific, but they're too specific for their own good, right? Or at least for our good, right? And so the question then is, can you make them sufficiently broad, but not not kill everything? So so the way we're thinking about it, and I didn't get a chance to explain the research we're doing, is that we're asking, can we, because the bacteria and the phage are in a constant arms race. The phage is always trying to get into the bacteria and kill it. The bacteria is always trying to evade the phage. And so they become over-evolved toward each other. And what is one of the important drivers of that specificity are the proteins at the tip of the phage that stick to the bacteria. And so that's where the phage gets its specificity from. Let me, again, maybe skip a couple of slides down. So there are these proteins of the phage that stick to the surface of the bacteria, right? And so one question is, so you can imagine the same phage infecting 2 different strains, but it just doesn't have those little proteins that allow it to go to a different, just wouldn't bind to it, right? And so one sort of synthetic biology approach is, can we swap out these proteins at the tip and essentially take the same phage but have it infect different types of strains, right? So then you don't have to go to nature and keep harvesting brand new phages every time, which can be a very time-consuming process. But essentially take a well-understood phage and just keep swapping out parts and make it go different ways. That's what we're trying to do. Sorry, yeah.
If you make it too broad, are you going to have to do a biome check of each individual so that you don't upset the individual's biome with a broader phage?
Yeah, I think that's a good question, and it's to some degree you can let me, all right, I'll use another slide as my aid. So this is the way I'm thinking about it, right? So you have some collection of microbes, right? And you know who the pathogenic guys are, right? By some lab technique or whatnot. And then you use phages that would specifically take out some number of species within that subgroup or evolutionary subgroup. And so the expectation is that it would be specific. I mean, there might be some differences across individuals, right? But they would be broad enough, but not super broad, that they would jump across. But again, we'll have to test this and see how far it goes. Sorry, gentleman in the back.
Could you say again what a phage is?
Can we see what a phage looks like? Is that the question?
What is it?
Oh, what?
What is it?
So a phage is a virus of that infects bacterial cells, and what it does is I'm describing one type of a phage. It binds to the bacterium. It shoots its DNA inside the bacteria, and then it makes many copies of itself inside the bacteria, and then it kills the bacteria. That's what a phage does. There are a few different flavors here, but that's the simplest version of it. So there's someone else that had their hand up.
Yeah, I was going to comment on that. To me, phage is a great example of the difficulty of terminology in biology, because it's just a virus. But they called him phage because it means eater.
Yes.
And you don't call animal viruses phages. You don't call plant viruses phages. That's what I worked on, was plant viruses long ago. And it's helpful when you know what the roots of some of these words are, because you can go, oh, that's just a virus. And yet we still call them the phages. We're going to have the phage meeting here on campus.
That's right.
In July, August.
Yeah, sometime thereabouts.
And if you're a bacterial virologist, you work on phages to this day. But it's confusing to people who may not study viruses.
Yeah, I mean it's a virus. They look in some phages look identical to human viruses. Some look different, but it's just a virus that infects bacteria instead of infecting human cells. Sorry.
I mean, there are also viral phages too, which are viruses that affect other viruses.
You know, I've read about that. I don't know a whole lot about it. That there are viruses that somehow infect other viruses, but I don't know the details of that.
So in nature, are viruses and bacteria enemies? Are they, I mean, forget the human. Is a bacteria and a virus both in the earth or in some? Are they going at each other all the time?
Yeah. So it's slightly more complicated than that, because really, so if the virus kills all the bacteria, it has no host, right? So there are elaborate computational models that tell you what that equilibrium is, right? It still wants to kill and make more copies of itself, but it cannot kill too many, because then it has no place to go, right? So there is a constant arms race, but there is a global equilibrium. So this, I don't know if this is making sense, but within a local niche, yes, they'll try to kill each other, right? But globally, there is an equilibrium between the number of bacteria and the number of phages, because there is a certain rate of evolution of the bacteria as it gets exposed to phages. They all undergo mutation all the time. Some mutation is just going to be beneficial to the bacteria, and then boom, it no longer is infected by the phage, right? And so that's the constant arms race, right? The phages will mutate 100 times faster than the bacteria. Yes, sir.
Is resistance to phage mostly the receptor, or is it internal?
It can be many different things. The receptor is one way, but there could be many. The resistance can happen at the entry level. It can happen once it gets inside. It's not able to make copies. It can happen. It makes copies, but is not able to get outside. It can just many different mechanisms of resistance.
If not, thank you very much.